Hello, good afternoon, dear students. Hope my voice is audible to you. Let me wait for 2 more minutes and the remaining people will join. Meanwhile, Anasuya, you can collect the uh, presentees and also the absentees. Please, all of the members, kindly mention your attendance in the chat box. Okay, dear students, now we'll start the session. It's already 3 minutes over. Please confirm whether my screen is visible to you or not. Yes, is it visible? Okay, thank you. Yes, thank you. Okay, dear students, in the last class, we have started the chapter marine drugs. So in the last class, I gave a little bit introduction to this marine drugs. These are the novel medicinal agents from marine sources. So this is also the one of the prime topic nowadays. So because of different sources are there for drugs like a plant source or animal source, mineral source. So similarly, one of the exploring area is marine source. That is, marine means those from drugs obtained from a ocean. Now we'll see. So already just introduction I covered in the last class, just so I want to skip all these slides. Then coming to this marine pharmacognosy, the word I want to tell here, marine pharmacognosy. So what is a pharmacognosy in general? That is a study of natural products in a simple language, right? So the study of natural products. So here the natural products, that those include from a plant source, animal source, and also those are from mineral sources, right? So including a these marine sources. If you are including these marine source, then it can be called as a marine pharmacognosy. So it is not a separate branch, so it is also a sub branch of pharmacognosy, which is mainly concerned with the natural occurring substances of medicine value from marine sources. Generally, these drugs are from obtained from marine species of bacteria, virus, algae, fungi, and sponges, etc. So, in case of Ayurvedic drugs or traditional drugs, those are from a plant sources. So, in plants, there may be these drugs are obtained from either from a leaf. 1 minute, the students. అవునండి, చెప్పండి. ఓకే. ఓకే. అక్కడ వెజిటబుల్ మార్కెట్ దగ్గరండి ఇవ్వండి. అక్కడ మీకు అక్కడ షాప్ ఉంటది. లేకపోతే వస్తారు ఉండండి అక్కడే ఉండండి. Yes, sorry, dear students. Sorry for the disturbance. So the marine pharmacognosy, it's not a separate branch. It is a sub branch of pharmacognosy. It's mainly concerned with the naturally occurring substances from uh, different marine sources, like those from a marine sources of a bacteria, a marine source of virus, algae, fungi, and sponges. Also, those of marine animals also here we can include. So that is the a marine pharmacognosy. It's not a separate branch, remember. And the next definition we want to remember is a marine drugs. So the drugs obtained from a marine organisms. So I already told in the previous slide, right? So not only as a microorganisms and virus and fungi, we can also include the marine animals like a shark, and also some of the cod liver. So cod liver oil, it is also obtained from animal source of marine. And also sodium alginate, it is a one of the fungus that is obtained from ocean. Also agar agar, and also chitin. All these are the different drugs. So those are obtained from a different sources of marine. So these drugs are very similar to the those can be applicable to the natural products. So here it is also same. These marine drugs are also same applicability for the treatment of a variety of disease and disorders of human beings. Now we will see this. Yeah. So what is the importance of these marine drugs? So because there will be a variety of applications are for there for this uses of these marine drugs. The first one, these marine organisms are potential source for drug discovery. See how these plant source as well as animal sources are there to find a new drugs. Similarly, there will be a similar concept are available for exploring of bioprospecting of drugs for these marine organisms, because the entire the surface is covered with 70 to 80% of water, and only a 20% left with the earth. So there may be a chance of different biodiversities and different bioactive compounds are there in ocean, so that you can isolate or you can find out a very different types of the compounds, marine compounds. Those are very useful to the treatment of a different types of the diseases. Like I already told in the last class, there may be some research also going on to find out some useful therapeutic agents to treat corona also. So here there will be a different animals like fish, sharks, snakes also. So there will be different types of vertebrates and invertebrates are also in and different biodiversities are available in ocean, so that there may be a chance of available in different drugs. So if you that the drugs means they are not directly available, so from these vertebrate animals and invertebrate animals, so you have to isolate from those sources, right? So this is a marine organism as a important source for drug discovery. Then second one is biological diversity in my marine environment. That means the marine environments are considered more biologically diverse than terrestrial environments. Terrestrial environment means outside the water, like here there will be a biological diversity is there. That is 32 different animal phyla are represented in across the oceans of the 33 recognized phyla. So the number of animals that is also more when compared to the terrestrial. So that is a one biological diversity is there. And another one, 15 different phyla are represented only in a marine environments, while only one is exclusively terrestrial. See, so except terrestrial, so we have find out different types of 15 different phyla we are find out in a marine environment. So out of only one we can exclusively found in a terrestrial surface. So this marine phyla is also contain functionally unique organisms such as a filter feeders and sessile organisms, so which have no terrestrial uh, counterpart. So this is also the another added advantage in order to find a biological diversity in marine environment. So this is another advantage of so why these marine sources that can be used as a drugs. So now that are the very important part of this our chapter. So here up to now what we are discussed, so there will be different sources are there for collection of the drugs from marine sources. Now how we can collect this sample? So because it is very difficult, so if it is a plant it is very simple to collect. If it is an animal, if it is very also simple to collect the that particular source, so from the plant and animals and minerals. But here it is a marine source, so how you can collect? There will be different types of the procedures and techniques are there for the collection of the samples from the ocean. That is from marine. So now we can see the what are those techniques. So here the collecting marine samples can range from very simple and also inexpensive to very complicated expensive. Samples from near or on shores are readily accessible via one is a beach combing. The one of the method that is now I will show what is a beach combing. So this is the beach combing. So this is a activity that consists of an individual combing or searching in the beach and the interdial zone, looking for things of value, interest, or utility. That means simply you can go to the beach and you can search for a what you want apart from what you want required. So from which you want to isolate the drug. This is a very simple technique. That is the a beach combing, one of the technique, right? Second one is the wadding. So you know I think you people are very well versed with this technique, wadding. So wadding with walk with effort through water or another liquid or viscous substances. That means so you have to walk through the water and you can use the either nets to catch the different sources. So that is the a simple technique of a wadding that is widely followed in our villages also, in small ponds and small rivers. Many of they are walking through these rivers and collect the different substances what they require. That is wadding. Second one is the snorkeling. So snorkeling is nothing but it is a practice of swimming on or through a body of water while equipped with a diving mask, a shaped breathing tube called a snorkel, and usually swim fins. So here, see, once you see the image, you can clearly understand. Here there will be a breathing tube that can be inserted, but there is no back end oxygen cylinder, right? So for this majorly a skillful drivers are required for this step in order to collect the sample. So that is snorkeling, there is a practice of swimming, right, with the help of a body, and they use some special type of shoe. Usually it can be called as a swim fins. So by using the swim fins they will dive through this underwater with the help of this mask that can be connected through the breathing tube, right? So this is another method. Snorkeling is one of the method for collecting uh, a marine source. Now we'll see the next this next one is the dredging. You know dredging means see here I am showing the one image, one large pipe. Okay, That is entirely almost this pipe is started from mid of the ocean to the and continues up to the beach. That means drugging means with the help of these large big steamers and big machines, they can drug into the ocean. So that there may be a chance of getting the material what they required. See, it is the nothing but it is the operation of excavating the material from a water environment. So with the help of these machines, they will collect the what they required. So like this, they are collected the what the material they required. They will collect the required material to this processing unit directly. From that, they are processed and then collect the required material. This is the one of the latest technique, drugging. Now we'll see the next method. Next one is a box corer. So this is a very very large equipment is there. There will be a single corer or double corer or multi corer is there. Here the picture I am showing, it is a the box corer. It is a marine geological sampling tool for soft sediments in lakes or oceans. Because some when we are go to the mid of the ocean, so there will be a soft sediment or a mud is there. So then it is not possible to dive into this mud, and also other techniques are not at all successful. So in those cases you can use the this technique called box corer. What they done, so it is deployed from a research vessel with a wire and suitable for any water depth. So it's simply inserted into the mid of the ocean, where they having a very rich for soft sediments, and so that they can be easily get into them those mud and collect the desired sample. Then, so the latest technique out of all those is the scuba diving. you know Scuba diving. So everyone can practice this step, right? So you see here, there will be one oxygen cylinder at the back. But in before other snorkeling technique, there is no oxygen cylinder at the back in case of snorkeling, right? So here in scuba diving, it is a mode of underwater where the diver uses a self-contained underwater breathing apparatus that is called as a scuba. What is a scuba? It is a self-contained underwater breathing apparatus. That apparatus is called scuba. So by using this apparatus, the person that can be dive into the ocean. So then it can be called as a scuba diving. Which is completely independent of surface applied to breathe the underwater. So you people very well you can compare the what we are discussed in the snorkeling technique. So here there will be a one breathing technique is there in snorkeling, but here there is no chance of trying and practicing those breathing techniques. This is entirely independent of surface supply of oxygen. So that means they have their independent connection to the surface. So they have a dependent and a one cylinder that can be carry at the back, that can be that can having a oxygen that can the diver uses to breathe the sufficient amount to survive. That can be called as a scuba diving. So by using this technique, the person may collect the source from this marine. So another one, this is a very much recent techniques, Aquarius. Aquarius is a it's a underwater laboratory. What we can call is a underwater laboratory. So what are the laboratories in our college is there? So those laboratories are on the surface, right? So here the Aquarius is one of the one of the laboratory that can be in underwater. So here the Aquarius is the name that can be given to. There will be as of now only three undersea laboratories are there in throughout the world. So one of such that laboratory name that can be given as a Aquarius. See, Aquarius. So one of the three undersea laboratories in the world and dedicated to the science and education. So this is also the one of the technique to collect the that is marine sources, right? These are the see. This is the before they can be placed into the placed underwater. So this picture that can be taken before it can be sent it to the ocean. So this is underwater, this is on surface, right? For our understanding point of view, I kept this image, right? So these are the different techniques, right? So I can repeat again. One is a beach combing. There is a simply you can collect from the beach source, and the second one is a wading. You can walk through the water and you can collect. And third one is a snorkeling with the help of these swimming and breathing tube. You can directly swim into the ocean. And another one is a dragging. Next one is a box scorer, and the next one is a scuba diving. And the last one and very recent technique is the using of this underwater laboratory. So these underwater laboratories that can be you can that can be called as a aquarius. So as of now I already told there will be only three laboratories are there in entire world. So it's very a costly process and very much very difficult to establish such a very huge this material into the ocean, right? Hope you understand. Are you following? Say yes or no. Mention short box. Okay, thank you, Divya. Yes, fine. Now we see the next slide. So there will be another technique that is also widely used by the scientists. That is submersible a watercraft, right? It's like a underwater vehicle. We can call it as a underwater vehicle. So here what they directly they sit into inside the this submersible, like a submersible watercraft, so that they can drive easily into the water, and there whenever they required they came out whenever they required. So they collect the required sample when that particular craft is reached to a specific point. So this is a submersible watercraft. So those are the different techniques of collecting the sample sources from ocean. Now we can see the next step. After collecting the sample, what is the next step? Isolating the compounds. So in order to chemical compound isolation, there will be a different techniques are there. Now we'll see the what is the chemical, what is the isolation, what is the different techniques. Just we can go for the outline. So for the isolation of biologically active compounds from organisms, several different steps need to be completed. The different steps required to obtain a biological active compounds are: first one is a extraction. See, so after you are collected that particular material from the ocean or a marine, after that we can subject to the extraction. Extraction means in order to get the desirable material from the out of that material, right? So because it is a marine source, like so it's having a different apart from the required component, it's also having unwanted materials like mud, shell, everything is there, so that we have to clear and we have to extract the required material, that is the extraction. Second one is a chromatographic purification. So this is a important step. So in order to get the a purified compounds, that means a chromatographic purification means either you have to use a HPLC technique or GCMS. You can use for the advanced chromatography technique for purification. The next one, dereplication. Dereplication means that means so after purification, in order to get the a further purification, this is one of the step. It can be called as a dereplication. That means a further purification of a what is the compound we required through the chromatographic technique. And last one, fourth one, not last one, fourth step is the a structural elucidation. Structural elucidation means so here we are getting the extract. Here in both these two steps we are getting the purification. After the purification we have to establish the that particular structure. That means if you are isolated a structure, so in order to we can give the complete structure. So what is this what is the bonds present in different locations, and what is the skeletons, what is the chemical structure, and everything that can be you have to elucidate. This can be performed with the help of IR IR technique and NMR technique and mass techniques. So these are the different spectroscopic techniques. By using those spectroscopic techniques, we have to find out the structure. Finally, bioassay testing. So the last step is the bioassay. That means now we can assay the for the particular activity, right? So these are the different techniques for the how the chemical compound that can be isolated after the sample is collected from the marine sources. So that can be the sample that can be collected by different sources, so we already seen. So this slide is entirely explained what are the different steps that can be involved in the isolation, that is extraction, chromatographic purification, dereplication, structural isolation, and also bioassay testing. So now here, there will be a the major problem in this case of these marine source is the supply issue. Supply issue. So it is a major problem. Yes, it is a major problem. Supply issue. So what happens? So we will see. A common problem that plagues drug development is obtaining a sustainable supply of the compounds. Is it possible to get the compound always from the ocean? So it is possible to get the compounds from the plant source. It's very easy because plants are is a part of our environment and grown very near to us, right? So it's very easy to get the plant material. But in case of the marine sources, so it is always not possible to go into the ocean, right? So there will be there will be a less chance of a sustainable supply. Compounds isolated from invertebrates can be difficult to obtain in sufficient quantity for clinical trials. Like let us say, if the compound you are isolated from a fungus or maybe a shell or a snail or maybe a specific creatures in ocean. So those are very much difficult to get to further to get the clinical trials, because we can get the very nano quantities, right? So the nano quantities, it is not possible for entire clinical trial team, so to get the clinical trials done very perfectly. So that those nano quantities not at all sufficient, because we are getting those nano quantities from a invertebrates. So those invertebrates are very very less in distribution. Then synthesis is an alternative source of the compound of interest. If the compound is simple, otherwise it is generally not a viable alternative. So the next step, so if the compound is very not available, so but then alternative to this that one is the a chemical synthesis. So this is also a major problem, chemical synthesis, because it is a marine source, so we are not at all completely not known the entire chemical structure and everything. So the synthetical procedure also we are not at all known. Then the next step is the aquaculture. So if the compound is very less in quantity, there will be a less we can another chance is you can maintain aquaculture is the another alternative if the organism is readily grown. Otherwise it may not be good sustainable source for a compound. So if the compound is very less, we can follow either chemical synthesis or maybe a aquaculture. These are the two alternative supplies. However, so the supply issue, it's a major problem in marine sources. But those supply issue we are not seeing plants, because the plants are very much easily available, and the cultivation and the collection is very much easy when compared to the marine sources. I can give here one example. Let us say the small quantity of the compound is usually found in from organisms makes this as a alternative even more expensive, because from the organisms sometimes we can get the very nano quantities of the compound. So because of that nano quantities, it makes what happens, the drug becomes more costlier, more expensive. See, here I am giving the one example. Here there will be a new drug that can be given as a ET743, that can be isolated from the tunicate. This is one of the one type of snail in the ocean. They yield only 2 grams per ton. See, 2 grams per ton. So మీరు ఒక టన్నుని ప్రాసెస్ చేస్తే దాని నుంచి వచ్చేది ఓన్లీ 2 గ్రామ్. So then in those cases, then what is the costly of the procedure? Then the drug becomes very costly. This would require thousands tons of tunicate to be grown and extracted to produce the kilograms of ET743 that would be required for the treatments of thousands of people. You can estimate. See, such type of problems we are seen in case of marine sources. So a large tons you can process, you can get a very small quantities as a yield. So in order to avoid this step, you can follow the other alternative techniques. I already told you, like aquaculture and chemical synthesis. Those are the different alternatives to this, to avoid this small quantities of this small quantities of the drug. Those are obtained from a marine sources. So the next is coming to this next slide: classification of marine drugs. So hope you people are following. Now we'll continue this classification of marine drugs in the next class. Just I will repeat the what we are learned today. We are very much learned today about this collection techniques, that is beach combing, wading, snorkeling, dredging, box scorer, scuba diving, and underwater laboratory, and also a craft, watercraft, that is a submersible watercraft. These are the techniques we are going to use for the collection of the sample, right? So after collection of the sample, this is the isolation technique, right? So isolation techniques include extraction, chromatographic purification, dereplication, and structural isolation, and bioassay testing. That's it. So that's it today from my end. So also we learned the supply issue with the help of examples. So then left with the classification of marine drugs. Now we'll see in next class, classification of marine drugs. Hope it can clear to all. If you're having any doubts, so you can ask in the chat box. Anusuya, you can collect the attendance, both the presentees and also absentees, and send it to me through WhatsApp. Okay, fine. So anybody having any doubts? Say something, yes or no. Okay, Vani, Durga, Anasuya. Fine. Okay. Let it be. Please wait. I will take the snapshot. Okay, dear students. We will see you next class. Okay. Anasuya, please post the attendance to me, presentees as well as absentees. Okay. Thank you, guys. We'll see you next class. Bye.